Hi, this is lecture 2 for PSYC1040, and today's lecture is going to be all about research design. And it's really about the broad kind of approach to research design that we will follow up with some more specific lectures in lectures 3 and 4. But anyway, let's go ahead and get started. Now, this is really about the fact that one of the big things that we care about in our psychology studies is how we design the study. There's a lot of thought that goes into it. Sometimes you might spend weeks or months designing a study, and how you design the study is going to affect a lot about the kind of data that you get and the kinds of statistical tests that you end up using. So, like I said, it's really important that we end up talking a lot about how to design a study properly. So first thing I want to do in this lecture is just talk a little bit about the research setting, about the lab, doing it in the lab versus the field. And then we'll talk about specific kinds of studies that could be done in the lab versus the field. So there's sort of two main research settings that are done in psychology. And the first kind is to do your research in a laboratory setting. And the second one is to do it in the field. So I thought it'd be kind of interesting to take a topic in psychology and just look at how two different teams of researchers looked at that topic, the same topic, basically, in the field versus in the laboratory. So in this example, we're going to look at attraction, how it is that we become attracted to another person. And the study I'll first talk about is actually a field study. So this is an experiment that was conducted in the field. And so here's the study. It was conducted in 1974. So it's an oldie but a goodie by Dutton and Aaron, some evidence for heightened sexual attraction under conditions of high anxiety. Now, in this study, what they were interested in looking at is whether or not people are more attracted to other people when they are currently aroused or anxious about something. So does the extra arousal that you have because of something else is causing that arousal get transferred on to this person that you find sort of attractive, and then you end up being even more attracted to them? And what they did to look at this is that they went to this famous bridge that's in Vancouver in Canada, and this bridge is still there today. I've actually visited it myself, but it's a narrow bridge that goes over a high river gorge. And as you walk across it, it sort of sways a little bit in the wind. And if you're anybody who's has any kind of fear of heights, it's very difficult. I personally suffer from a fear of heights, and when I went across this bridge, it took me a long time because I was like holding on so fiercely to the ropes as the bridge starts to sway. So anyway, what they did in this research is they tested people who happened to be going across this bridge or across another bridge, a low bridge. So they had this high bridge, and then they had another bridge just a little bit down the river that cars go across. And it's a nice solid concrete bridge, and it's nice and low. And normally anybody walking across there wouldn't really feel any kind of anxiety about anything because of the lowness of that bridge. And so what they did was they had an independent variable here, whether the participant is on the high bridge or the low bridge. And then as this person's going across the bridge, they run into another person, okay? Now, all of the participants were males, and what they did is they had a female confederate or a female assistant in the study who acted like she was just some normal everyday person who walked up to the participant as they were crossing the bridge, all right? And what they did was they asked that confederate would just make contact and ask them to answer a couple questionnaires. So she'd come up to me and said, "Hi, I'm doing a little survey on creativity. Would you mind answering some questions for me?" And then she gave them, the participant, her phone number and said, "If you have any questions about the study, feel free to call me later." Okay, so that's the thing. So this woman walks up to you as you're crossing the bridge, asks you a few questions about creativity, and then gives you her phone number if you're a male participant. And the idea then was we have two different dependent variables. The first one is whether or not in that open-ended questionnaire on creativity that she asked questions about, they brought up anything sexual. Did the person flirt or make any sort of sexual comments on that creativity? And then on that creativity questionnaire. And the second dependent variable is whether or not the participant called this woman later. Did she actually do it? Well, it turned out that the people who were in the high bridge condition, those men that were in the high bridge condition, were more likely to bring up sexual content in their answers about creativity. And they were 8 times more likely to call that female confederate at a later time than when compared to the people on the low bridge. So those men who are on the high bridge, 8 times more likely to call her than those guys on the low bridge, and they were more likely to bring up sexual content in their questionnaires. So that's confirmed their hypothesis that having that heightened anxiety about being on a high bridge actually causes you to transfer this to that attracted person, and you feel even more sexually attracted to her. That was what they used as their evidence. Okay, so that's a field study, and you can see how they ran that study that way. Now here's another example of a study, and this one is a laboratory example. It's more recent, it's in 2008, and it's actually an fMRI study. So it's using functional neuroimaging in the laboratory to look at see whether or not attractive people are more rewarding, okay? So there's strong evidence that they say in their paper that we prefer attractive people over unattractive people, right? So we already know this from the literature that people like to look at attractive people more than they look at unattractive people. Even infants do this. And there's also evidence that men value physical attraction in the opposite sex more than do women on surveys and so on. So what these researchers hypothesized is that they would find a sex difference in brain areas active in reward circuits. And these particular reward circuits in the brain that they were interested in were like the nucleus accumbens and the orbital frontal cortex, which are also active, for instance, when people are addicted to drugs or taking a drug. So they wanted to see, like, do those areas become more activated when you're looking at somebody that you find physically attractive? So what they did is they had men and women in this study who judged the attractiveness of 90 faces of the opposite sex while they were in an fMRI scanner. So you're lying there on your back in an fMRI scanner, staying very still, and you're being shown one picture at a time, and you rate how attractive you think this person is. And they're all opposite sex from the sex of the participant. And when they looked at the ratings, these are the behavioral results, you can see that there's actually no difference between the way men and women really rated these pictures. They're equally likely to use, you can see this tells us how fast they responded, the number of responses they made, and the ratings of attractiveness are 1, 2, 3, 4. So they rated them on a 4-point scale where 4 was the most attractive, 1 was the most attractive, 4 was the least amount attractive. And the only thing that they really found here was that women were more likely to choose not attractive at all than men were down there in the lower right corner. So they didn't really see much in terms of the ratings between men and women. But when they looked at the brain activity of these men and women, they found that activity in the left and the right nucleus accumbens was greater in the men and the women for more attractive targets. Okay, so this one area of the brain, the nucleus accumbens, which is considered part of the reward circuit in our brains, was greater for both men and women when they were looking at targets that they viewed as being attractive. The orbital frontal cortex, which has also been involved in things like addictions, was different only for male participants. And so it didn't seem to be different for attractive and unattractive targets for the female participants, but it was for the male participants. So you see, here's another study many decades later, also interested like in an experimental way here of looking at how people respond to other people in terms of attractiveness. But this study obviously was done in the laboratory. So we have some studies that are done out there in the field, outside of the lab, outside of the psychology department, outside of the university, and we have others that are done in the laboratory. And there are pluses and minuses for both of them, and we'll talk more about those as the weeks progress. Now, under the context of either doing it in the lab or doing it in the field, there are 4 main research methods that we can address in psychology here at this particular point. And those 4 main research methods I'll cover in today's lecture are the experimental method, the quasi-experimental method, the survey method, and the case study method. All 4 of these have had an important role in the development of psychology as a science, and so we'll take each one of them and just give a brief introduction about each. The first one we'll talk about is the experimental method, and I will be actually talking about this all next week in the next lecture, but the experimental method's got the key feature of manipulating the independent variable. So you remember, the independent variable is the variable that an experimenter or a researcher can control, and that means that he or she can assign people to one condition or the other at will. They have an ability to do that. And how they do that is they typically are going to use random assignment. So random assignment is how you manipulate it. So you decide who goes into which condition using random numbers. So like I said, a key feature of this is to use randomization. And if you ever hear someone talk about a between subjects design, this means that each participant can be assigned to one of the conditions that are in the study, so just one. We'll also talk next week about a within subjects design. This would be a case where a participant goes through all of the different conditions, but for now, let's just keep it simple. In a between subjects design, let's say you have the experimental group and you have the control group. Randomly assigned means that you could use a coin toss or a random number generator or a table of random numbers to look at, to help you decide whether or not they should go with that one condition or the other. I'll just go ahead and throw out that if you go to this site called random.org, this website is really useful. It's got many tools for generating random numbers. And as you go down through the different choices on their webpage, you'll find that they have a free set of tools that you can use to do randomization. So you can do things like choose 100 numbers and randomize the sequence, or you could just pick the numbers 1 and 2 and create a whole bunch of random orders of 1 and 2, so that as participants come into your study, you could look on the next line and say, oh, you're going to go into 2. Oh, you're going to go into 1. Or if you have 5 conditions, you could go ahead and have it pick numbers from 1 to 5 randomly. So it's just a nice website that actually has very good algorithms for creating random numbers. So in an experimental design, like I said, you're going to randomly assign people to the different conditions. So here's an example of an experimental design. This is just completely fictional. But let's just say we randomly assign 100 students to different kinds of learning for one semester. So they're going to be randomly assigned to be either in Group B or in Group A. In Group A, what's going to happen is they're going to learn the material for that particular subject completely self-taught. A whole bunch of material has been put online, and the students are told, go through that material and teach it to yourself during this coming semester. Group B is more of a traditional kind of test, or a traditional kind of teaching format, where they're going to go to watch lectures and have assigned readings throughout the semester. And so what this would mean is that we'd randomly assign people to Group A or Group B using like one of those tools on random.org. So you're going to either go to Group A or Group B. Flip a coin. That's another way to do it. Flipping a coin is considered randomization. So you can flip a coin heads. That next person in the study is going to go into Group A, or they're going to go into Group B. But the point is that you didn't decide, you didn't just say, hey, I like you, you're going to go to Group A. I don't like you, you're going to go to Group B. You actually use randomization to assign them to those two groups. And then what you could do is have a dependent variable at the end of the study. So for instance, at the end of the semester, they could look at their performance on an exam. So everybody completes the same exam, and then we could find out whether or not Group A or Group B did better using self-taught versus lectures. So I think what you can see is the virtue of random assignment is by me not systematically saying like all of the smart people go into Group A and all of the dumb people go into Group B or anything like that. I'm going to go ahead and just use randomization, because whatever I see there at the end then really tells me a lot about whether or not the type of learning affected their performance on the exam. I can feel very confident about what I learned there at the end on the performance on the exam, about whether or not it matters if you're self-taught or you go to lectures. And of course, another possibility could be that there's no difference between the two groups at the end, that they have essentially the same scores. And so that would tell us that it didn't matter which one worked. All right, so that's an example of an experimental design. And like I said, we'll go through this in a lot more detail at the next lecture. The second kind of research method I'll talk about is the quasi-experimental method. And the quasi-experimental method is very frequently used in psychology because it's like an experiment, but what we call the independent variable isn't really being manipulated by the experimenter. And it's because it's either impossible to do this or maybe it's unethical to actually manipulate it. But for all intents and purposes, we go ahead and pretend or act like the study is an experiment, even though we didn't actually get to manipulate the independent variable. So the examples of this would be things like you could look at whether or not a person has a brain injury. So you could have one group that has a brain injury, the other group that doesn't have the brain injury. You see, it would be unethical for you to randomly assign people to brain injury and then injure their brains, or if they're not injure their brains. But that would be an example of using random assignment, but it would be unethical to do that. Another possibility would be to look at age, like old versus young, and it's basically impossible to go ahead and manipulate people's age or what their nationality is. So again, it's important to keep in mind that for all intents and purposes, though, quasi-experiments look a lot like actual experiments. And there's a key reason why I bring that up is because sometimes the researchers just seem to forget that they didn't actually run an experiment, that they still are using a quasi-experiment. And let me show you what I mean. So for example, let's say that we want to compare smokers to non-smokers to see if smoking affects your number of visits to the doctor and how healthy you are. So what you do is you go out and you try to find 50 non-smokers and 50 smokers, okay? And group B becomes the smoker group, and they're defined by, on average, they have about 30 cigarettes per day. And the group A is 50 people that you found that are non-smokers. They had 0 cigarettes per day. Now notice that this isn't randomly assigned, right? I mean, sure, it looks like I randomly picked 50 people in group A or randomly picked people in group A and B, but I didn't actually take each person, flip a coin, and decide whether or not they were a smoker or a non-smoker. They already, before my study began, were a smoker or a non-smoker, and I'm making use of the fact that they are a smoker or a non-smoker. But anyway, I have those two groups, and so I want to compare them to see if the smokers have worse health than others, than the people who don't. And so what I do is I measure, for instance, the number of doctor visits they have in the next year. Okay, so I look at the number of doctor visits that the smokers have, the number of doctor visits that the non-smokers have, and then I could compare that and see if there's a difference between the smokers and the non-smokers. And so, for example, at the end of the study, I might get some results that look like this. Where on the y-axis, I have the number of visits to the doctor per year. And you can see that I have those two groups. The number of cigarettes smoked per day is really just, it's not really what they are. It's just saying that group A was the group that doesn't smoke at all. Group B, on average, smokes 30 cigarettes per day. But it looks like the people who smoke go to the doctor more than the people who don't. Now, can I say that I know now that smoking makes you go to the doctor more in the next year? I can't say that. And the reason why I can't say that is that I didn't actually randomly assign them to the two conditions. I might have a problem that there's some other factors that are different between people who smoke and the people who don't smoke. Let's just say, for example, that maybe people who are smokers also tend to already have other health problems. And so they were already having health problems before they became a smoker. And now when I look to see how they are functioning over the next year, the reason why they're actually going to the doctor more isn't because they're necessarily smoking. It's just because they were already in bad health when they began to smoke. So I don't have that same sort of confidence that I had when I was doing an experiment where I could randomly assign them to the two conditions. All right, let's move on to our third research method, which is the survey method. Now, the survey method could also be called other things, and this would include doing observational research or conducting a correlational design. All of these things, when I say the survey method, are pretty much these same things, so observations, correlational designs. The idea behind surveys is that this allows the researcher to gather information about a number of different variables, maybe 100, 20, 100, 200, 300 different variables all at once, to study the relationships between those variables. So I get a whole bunch of dependent variables, and I measure height and age and attitudes towards Australia and whether or not they smoke and all these things about them, okay? And then I look at relationships between those variables. That's the survey method or the correlational method. All of those relationships that I would be looking at are actually correlations, where I can see if there's any kind of association between how old you are and how much you spend time on Facebook, for example. So this is a very common thing that we do in psychology. We conduct a lot of surveys, and those surveys, when they just collect a lot of dependent variables like that, really are just correlational studies. The downside to this, then, it's difficult, if not impossible, to determine the causality. All I can really tell you is that a couple of variables have a relationship between them, but I can't really tell you whether or not the variable is being caused by the other variable. But like I said, it's a very common thing. And at the fourth lecture, we will be spending that particular lecture on the survey method in more detail. Now, just to stop a little bit before I go into the fourth method, I just want to recap a little bit about hypotheses and kind of focus on a little bit the difference between these kinds of methods that we've been talking about so far. So you might remember from the previous lecture that I said that a null hypothesis is when we state that there's no relationship between the independent variable and the dependent variable. And then the alternative hypothesis, or a research hypothesis that we're hoping to show, is that there is a relationship between the independent and the dependent variable. Now, those statements that I have there are really clearly for experiments, right? Because experiments are the only ones that truly have an independent variable and a dependent variable. You could also, though, make null hypotheses and alternative hypotheses about relationships between dependent variables. So you could say that there is no relationship between dependent variable 1 and dependent variable 2. And then if you reject that, you could say that there is a relationship between dependent variable 1 and dependent variable 2. So let me show you what I'm talking about here. So let's say, for example, that I'm doing a survey or an observational design here. Again, it's correlational. And all I did is I asked my participants, how many cigarettes do you smoke per day? And how many times have you been to the doctor in the past year? And let's say I find when I plot the individual participants, each little dot there represents an individual person, that the more cigarettes a person smokes, the more likely it is that they did go visit the doctor in the past year, right? They have more visits. So like that person who has 40 cigarettes a day or 35 or 32 cigarettes a day, they're going to the doctor much more often than people who have somewhere between 0 and 10 cigarettes per day. So why I say that's support for H1, this is the support for the research hypothesis. This says, oh, there is a relationship between the number of cigarettes per day and the number of doctor visits per year. Whereas the null relationship, or the null hypothesis, would say that there isn't a relationship between the number of cigarettes per day and the number of doctor visits per year. So in a case where I would have the null hypothesis, I'd be stating there is no relationship between the two variables. I'd see it like this, where you can see that it doesn't matter how many different cigarettes you've had per day, you have randomly just anything happening in terms of the number of doctor visits per year. There's no relationship there. So in correlational research, they like to see correlations over there on the left, because that shows us then support for our research hypothesis, that we actually are showing evidence that there is a relationship between the number of doctor visits a year and the number of cigarettes per day. Now keep that in mind and go back to look at quasi-experiments. Now you remember this is the graph that I showed you, the figure of what it would look like if I did a quasi-experimental design, where I just grabbed 50 people who don't smoke and I took 50 people who do smoke, and I go ahead and look at the number of visits to the doctor they have for the next year, okay? What you can see there is that I made it look like, from this fictional example, that the people who have like around 30 cigarettes per day on average, that group goes to the doctor a lot more than the people on the left. But can I say that I have a cause there? No, I have no causality here any more than I do on the graph on the left. They are both correlational studies in the sense that if you look at it, those are actually the same dependent variables. Down at the bottom there, the number of cigarettes smoked per day was how I determined the two groups, 0 or 30. And then over on the other axis, the y-axis, I have the number of visits to the doctor per year. I'm really just showing a correlational research on both of those graphs. So both the quasi-experimental design and a survey design don't help us understand causality any more than they just show us that there's a relationship between the variables that we're interested in. So remember that even if you make a graph that looks like the one on the right, if it's a quasi-experiment, it doesn't have any more evidence about what's causing what than the correlational design does. Now, as I said, I will come back to this again in 2 lectures. We'll talk more about survey research, and we'll understand a little bit more about why that is, why we can't really understand the causality. But for now, just get it into your head that really it's only the experimental method that we've talked about so far where we can really start to get a sense of whether or not something is causing something else. Now we go to the 4th research method, and I'll just really briefly touch on this because we don't really talk too much about this method in this particular course, but you might find, for example, if you take any kind of courses that have neuropsychology in them, that you'd hear more about this, the case study method. Now the case study method is what it sounds like. Usually what you do in a case study is you find 1 or maybe 2 cases of somebody who has something exceptional about them. They might have had a particular brain injury. They may have an ability that most people don't have. For instance, maybe they smell things from a very great distance away, so they have a really acute smell ability. So there's something unique about this person, and so you want to study them, and you'll study them intensively. So in the case study method, what a researcher usually does is they take that one person and they subject them to many different tests. They study them in many different ways, but they only have that one person or those two people. Now, there are some famous case studies in psychology, and I'll just mention two of them just to get a sense of what this is like. One of them is this guy, little Albert. Now, little Albert was a young child here, a few months old. In 1919, the behaviorist John Watson actually was interested in understanding behaviorism and how it explained emotion. And what he said was that John Watson believed that there were just 3 unconditioned emotional responses that all babies are born with. That is, he thought that all babies were born with these 3 basic emotions: fear, rage, and love. And so what he suggested was that all the other emotions that we have as adults come from those 3 basic emotions: fear, rage, or love. And we do this all through conditioning. So he wanted to show through Pavlovian conditioning that we could be conditioned to have these responses to new kinds of stimuli. So in 1920, he ran this experiment on Albert, this one poor baby, with his grad student, Rosalie Rayner. And what they did was they basically had little Albert there interact with different kinds of things. So, for example, they would have him touch a nice furry animal or a stuffed animal or some other kind of toy, and they would see how happy he looked and so on. And what they found was that he generally had normal kinds of responses to all these different sorts of stimuli. But then what they wanted to do is to see if they could go ahead and change his response to those normal stimuli through a conditioned response. So, for example, they would hand a little Albert a white rat. So this little furry white rat is presented to Albert. And so Albert reaches out to touch it because it looks like nice and soft and furry. And just as his hand would touch the animal, a bar would be struck immediately behind his head. So Watson would be standing there behind him and go bang really loud behind the head, and would basically surprise little Albert and make him scared. And he would just jump violently and fall forward, according to the article, burying his face into a mattress. But he wouldn't cry. He just seemed really upset. And then he reaches forward again to try to touch the rat, and again the bar was struck, and again the infant jumped violently, fell forward, and began to whimper. And in order not to disturb the child too seriously, they stopped the test for 1 week. And then a week later, they present the rat to Albert again. He keeps his distance, and anytime he gets anywhere close to it, they would go ahead and go bang. Okay? So what they basically were showing is that they could condition Albert to have fear towards this nice furry white creature that he originally had no fear of. And so in the article they say, the instant the rat was shown, the baby began to cry. Almost instantly he turned sharply to the left, fell over on his left side, raised himself on all fours, and began to crawl away so rapidly that he was caught with difficulty before reaching the edge of the table. So what you can see is he's really scared. And what they then after that did was they went ahead and showed him some other white furry stimuli like a rabbit, a dog, a seal coat, some cotton wool, and a Santa Claus mask. And each of these, when they were presented to Albert, would cause the child to also cry and get upset. So this showed that he had sort of generalized fear to anything that was white and fluffy. So he kept showing these pronounced fear responses, and then they were done. They were done with the experiment, and that was the last that Watson and Rayner saw of little Albert. They let him leave the hospital without trying to decondition these fears that they had produced. They added in their paper that they would have done this, but unfortunately the parent of this child just took the kid away, and so they were never able to fully decondition little Albert. Now, poor little Albert, we consider this to be sort of an unethical experiment today, But at that time, that did tell us a lot about how conditioning works, and it gave evidence then for Watson's theories about how emotions are acquired and conditioned. So you see, it's just one case that's thoroughly tested, and then we learn a lot about something. I'll give you one more example, and this is HM. This is also talked about in your textbook. HM was a man who had his hippocampus removed in 1953 by a neurosurgeon as a way to treat HM's severe seizures. Now, he's called HM because those were his initials. You can actually find his full name in the textbook and other places, but he's usually just called HM. And then the thing that happened is that after he had recovered from this, his seizures did go away after having this surgery. But interestingly, the neurosurgeon and his physicians noticed that HM could no longer make new memories, that he couldn't remember anything that happened to him since he had had the surgery. And so you might introduce yourself and say, hi, I'm Jack, and then he wouldn't be able to retain who you were a little while later. There'd be no memory of him ever meeting you before. So they referred him, HM, to some psychologists. The first group of psychologists were Penfield and Milner, and then another psychologist named Corkin and her colleagues worked with HM. But they spent 55 years actually studying HM and looking at these kind of memory problems they had. And they learned a lot about memory, about how memories were created and how the hippocampus seems to be an important part of that memory, all from this one patient, this one case example of somebody who had had his hippocampus removed. So again, very intensive studies, many papers done about this one individual because he was exceptional. Exceptional in the sense that he's one of these poor, unfortunate souls who had his hippocampus removed to have these seizures treated. And they don't do this anymore, but psychology sort of benefited from the fact that poor HM couldn't recall things after this. So those are examples of case studies, and you do run across them from time to time in your psychology courses. But like I said, we won't be talking about them much more in this particular course. Okay, now the last topic that I want to focus on in this lecture is about dependent variables. Now, dependent variables, again, are these things that we measure, and we think that they're usually going to be changing as a result of changes in the independent variable, or we think they're related to other dependent variables in our survey. So let's think about that. So a dependent variable is something that you're measuring about somebody. It's variable. That means if I go from person to person, everybody's going to have maybe a slightly different response on that measure. So for example, I could ask you, how tired are you right now? Everybody who's watching this lecture, and you could give me a rating from 1 to 5. Like 1, I'm not at all tired, to 5, extremely, could fall asleep right now. And as we went around and asked everybody who watched this lecture to give us their answers, we get some people give us 1, some would give us 2, some give us 3, some give us 4, some give us 5s. So this variable called tiredness has a lot of variability. There are 1s, 2s, 3s, 4s, and 5s across the entire set of people that I could go ahead and measure how tired they were. So that variability is something that we're really interested in, the variability that happens on that dependent variable. And if you look at it more carefully, you can think of it this way. What I'm doing here is I'm saying, all right, let's take experiments and quasi-experiments and look at the variability that happens in the dependent variable. All of these different answers I could get, 1, 2, 3, 4, and 5. Where are they coming from? Well, over on the left, you can see that if I'm running an experiment, I'm going to be interested in my independent variable. So it could be that the independent variable is causing a change on my dependent variable. So that is probably why I'm running the study in the first place, that the dependent variable is changing because of changes in my independent variable. So that could be the main thing I'm focused on in the experiment. But there are also other things that could explain why I get variation on the dependent variable. And these include things like measurement error, individual differences, and situational variables. And you'll notice that all of those I put down as random variables. They're called extraneous or random variables. So the thing over on the left are the independent variables, and the dependent variables being affected by the independent variable. That's what I was hoping to show in my experiment. But another thing that could also be explaining why I'm getting a variety or variability on my dependent variable are these random variables. Things that are extraneous, that are causing the dependent variables to have their variability. And so measurement error would mean that perhaps I'm just measuring my tiredness not in a very precise way. Maybe that 1, 2, 3, 4, 5 is just a very imprecise way of measuring how tired people are, and maybe people don't even realize how tired they are. And so some people are more aware of it than others. And so the problem is that I'm getting a lot of error just in the way I decided to measure my dependent variable. So that could partly explain why I'm getting different answers from different people. Another way that I could get lots of variability in my study could be just from individual differences, that people have different chronic different ways that they respond to things. And it could be, for example, in tiredness, that maybe some people are just tired all the time, or they just have a habit of always saying that they're tired even when they're not. And so the individual differences wouldn't really have to do, the differences on tiredness don't really have to do with my independent variable. They could do to more with just like individual differences that happen between people's personalities or their backgrounds or whatever it is. So that would be considered another extraneous or random variable. And also, finally, could be situational variables. So when I look at how tired people are, maybe I'm trying to show that I can make some people tired and other people not so tired, but maybe there was something else going on I didn't know about. Like, for instance, I tested a bunch of people during a time when they had some exams or some other huge crisis was happening in their lives. And so there was a situation that was causing some people to feel tired and other people not to feel so tired. I wasn't aware of that. And it's creating a problem in my experiment because I'm getting variability now from this situational variable. So these are all other sources for the variability in my dependent variable. So all of those things, the measurement error, the individual differences, the situational variables, those are all things that are affecting the dependent variable. But the other thing that I have here on my little picture is something called confounding variables. And confounding variables are variables that vary the same time as the independent variable. So that means that whenever the independent variable switches, like somebody goes from condition A to condition B, or they've been assigned to the treatment group or the control group, that there's another variable that also happens at the same time when I assign people to the treatment group or the control group. It means that I have a confound. And I'll give you an example of this in just a moment. But confounds are really something that can ruin your experiment if you don't pay attention to it. I'll just say, by the way, before I go into more detail about confounding variables, that one of the things that we spent a lot of time on in our design of our experiment is trying to design our study to have control over those other sources of variability. So we try to do what we can to reduce the random variability, to reduce the influence of individual differences, and to reduce the confounding variables. Those are all things we're trying to design out of an experiment or out of a study. The more we can do that, the more confident we can feel we understand the relationship between our variables. So like I said, one of our aims is to reduce, this variability, and that would be to reduce confounding variables so that you can interpret the relationship between the IV and the DV. And then you also try to minimize any kind of random variability, again, to improve your chances of finding a relationship between the independent variable and the dependent variable if one exists in an experiment. So let's go back and talk about what a confounding variable would be then. It'll sound pretty straightforward, but a lot of people kind of get it confused after a while. Remember that it's always connected to the independent variable, these confounding variables. So to look at confounding variables, look at this example. Let's imagine that we have our two groups again. We have a group A and a group B. And what we're doing is we're going to look at the effect of getting no sleep on reaction time. Okay? So group A are 50 people who get no sleep one night. Group B get 8 hours of sleep. And people have been randomly assigned through their coin flip whether they get into group A or group B. They've all agreed that they do this study. And then what we do is we measure their reaction time, how fast they can press a button to different lights that appear on the screen. The problem is that we go ahead and measure group A in the afternoon. So all of their reaction times are done in the afternoon. Group B, all of their reaction times are measured in the morning. Why do we have a confound here? The problem is that now what I've done is I've made the time that I measure the dependent variable happen in a way that completely is in sync with what the manipulation of the independent variable is. Notice that everybody who doesn't get sleep gets tested in the afternoon, and everybody who does get sleep is tested in the morning. This is like something I wouldn't want to do, but I'm just giving to you as a good example of a bad practice. Like I wouldn't want to have this happen, but what I've just done, because I didn't think about it very thoroughly, is I created a confound. And my confounding variable is the time of day in which the reaction time is measured. Because now the problem is that if I do see a difference between group A and group B, I don't know if group A is different from group B because they didn't get sleep, or is it because their reaction time was measured in the afternoon, whereas group B could be because their reaction time was measured in the morning rather than the fact they got 8 hours of sleep. So you see, a confounding variable happens when it systematically varies with the independent variable. So everything else I've been talking about, like individual differences and situational variables and so on, those are all things that are considered random variables. They're things that we try to eliminate. Confounding variables are things that we sort of like just blundered through in our experiment. We didn't do a very good job, and we ended up doing something where another variable systematically varies with our independent variable. So keep that in mind, that a confounding variable is a variable that can be classified that way only when it systematically varies with the independent variable. We'll try to give you some more practice with this in the next couple of weeks to see if you can have a better understanding of the difference between a confounding variable and a random variable and so on. So that's all I have in this particular lecture. The next lecture will focus on experimental design in much greater depth, and we'll talk about things like within subjects and between subjects designs and so on. Thank you.